Our next speaker is an external speaker. We are delighted to have Dr. Michael Washburn from the Stowers Institute of Biomedical Research, Kansas City. He's the director for proteomics in the Stowers Institute. Dr. Washburn's name is synonymous with the widespread application of MudPIT technology. He has worked with Professor John Yates in the West Coast prior to his arrival in Kansas City. His focus areas for his current research include quantitative proteomics and proteomic technology development, with notable specific applications being study of dynamics of multi-protein complexes, including transcriptional regulatory complexes, and also a growing interest in his lab is probabilistic assembly of protein interaction networks. With that being said, the title of his talk today is Using Normalized Spectral Abundance Factor to Visualize Protein Complexes and Protein Interaction Networks. Welcome once again, Dr. Washburn.
Thanks so much. Well, thank you all for putting together an inspiring day. This should be really entertaining. I love one-day symposium like this on topics that you're really passionate about. It should be really wonderful. And thanks to Mark and Rob for setting a prelude. Let me skip through a couple slides a bit faster. So again, this is the title of my presentation, and mostly I will be talking about, it's kind of like a third of the type of work that goes in the group. We do a fair amount of mechanistic transcription and then some fundamental analytical chemistry on spectral counting. But a major effort is now going into protein complexes and interaction networks. So there's four parts, where we'll talk about this normalized spectral abundance factor. And I'll give a couple of presentations on approaches that we use to look at protein complexes, a quantitative analysis of a complex called mediator, and then our efforts to assemble protein interaction networks in a probabilistic manner. And finally, a very recent study on where we're starting to look at, and we're really interested in dynamics. So this is a deletion network that we looked at to see what impact that has on a very small interaction network, but it's an approach that we're using very widely now. So as Yuri said, my group is all MudPIT all the time, and most of you have probably seen a slide from similar to this from John Yates in the past, but so we're going to be using these chromatography-based columns to load protein complexes on and analyze everything in this fashion. And so there's maybe 40 of these little stations around the laboratory to constantly prepare these little MudPIT columns. MudPIT basically is a kind of a pseudo three-dimensional chromatography approach where you have low and high acetonitrile and a semi-volatile salt so that you can, over the course of 10, 20, however long you want to do it, elute peptides in the mass spectrometer for analysis. The key aspect, obviously, for all of this is this usage of taking experimental tandem mass spectrum compared to theoretical tandem mass spectrum to get your actual results. We use Sequest. There's plenty of other approaches out there these days. But this is kind of the fundamental aspect of the quantitative proteomics approach that we use, which is spectral counting. So we're going to use the number of actual tandem mass spectra that correspond to a particular protein for all of our quantitative analysis. So here's kind of this is a 6-step MudPIT analysis of something. I don't know what anymore. But you can kind of imagine the idea that, so here's perhaps one spectrum from one peptide that showed up. That would be a very low abundance protein. And then in this case, since we're using a repeat count of 2, we're getting 6 tandem mass spectra. There's going to be a medium to low abundance protein. And there's this other peptide here that we get very many peptides, 112 across the course of analysis, and that's going to be a high abundant protein. So we're using this range of spectra and this range of peptide matches to do our quantitative analysis. This is a data set from other work in the laboratory looking at RNA polymerase II that I won't talk about today. But the idea being here, this is from DTA Select. So here we have a component of RNA polymerase II where there were 9 unique peptides. Some of them show up 2, 3, 4. This one showed up 33 times, 45 times, but 115 spectral counts. Here's another protein from RNA polymerase II where we have 12 unique peptides and 329 spectral counts. This one being one of these peptides that people like to call proteotypic peptides that are massively overrepresented in many data sets. Here's another protein, RPB4. We have 15 unique peptides, but 470 spectral counts. So we're going to use these spectral counts as our quantitative tool. Now, people in the field have noticed, and we're not unique in this, that longer proteins have a tendency to have more spectral counts than shorter proteins. And there's been a number of publications on this in the past. So you have to take into account the length, molecular weight, or some people like to use observable peptides. We don't use the observable peptides because we oftentimes use enzymes other than trypsin for doing certain types of studies. The other thing that we noticed, and everybody notices, is that when you do multiple runs for statistical analysis, the identical number of spectral counts for replicates is very difficult to achieve. So we'll have these slight increases or slight decreases kind of on a systematic, uniform fashion. And I spend a fair amount of time reading, say, the genomics literature about how they're analyzing, say, Affymetrix or 454 sequencing or Selecta sequencing data sets these days. And in the genomics community, in microarray world, Affymetrix world, there's always been normalization approaches to account for these changes in intensities from replicate to replicate to replicate. And this is something that I think is really important for quantitative proteomics in general that's very widely underutilized, and it's essential for statistical analysis because of this variation that you're going to get from biological replicate to biological replicate, or even technical replicate to technical replicate. So our version of dealing with this is something called this normalized spectral abundance factor, where the idea is you have an indicator of abundance. Longer proteins have more peptide identifications, and then we have this variation from run to run in the MudPIT analysis. And so here we're going to have the spectral count divided by the length for any given protein K, divided by the sum of all spectral count divided by length for all proteins identified in a sample. So this is this normalization, this denominator portion, that's quite important for bringing everything kind of together and making things more uniform. Now we're going to get this value. It's going to be between 0 and 1 at the end. And effectively, what it represents is like what percentage of a given sample came from a particular protein. So how does this help? So here's 3 different replicates of RNA polymerase II sample where we have one had 13,000 spectral counts, one had 21,000 spectral counts, one had 18,000 spectral counts. There's pretty big variance there across 3 different biological replicates. Upon normalization, though, we're getting 0.56, 0.57, 0.60. So this is a really important step for us to be able to do good statistical analysis at the end of the day. And also the normalization allows us, which we'll see later, to compare the abundance of protein A to protein B. We're not just talking about abundances of individual proteins. Now we could take into account all the data that comes from a data set. So an early application that I always talk about, because it's something that I think is really important going forward in the field, is to look at protein complexes in a very quantitative and dynamic fashion. And we use this approach for a lot of ongoing work in the laboratory. So we're going to be looking at mediator. And Rob did a really nice job presenting this actual workflow. The only thing that I'll add to this is that in my laboratory, we also run all these affinity purifications. We usually do non-transfected cells. Sometimes we actually have the epitope itself. And we're going to use silver staining gels very frequently. We kind of use them as a quality control to make sure somebody actually has a sample. Or sometimes, especially with vast overexpression, you'll have this giant silver streak with a big blob in the middle. And that tells you a lot about how the sample is going to be and how good your data analysis is going to be at the end. But so we use these for quality control, but then we run MudPIT on everything. I've always been curious where this slide came from. I got a postdoc who gave it to me, but they're like the same little pictures have. I have no idea where they all generated from. Possibly. You know how slides, they turn out, they can become a life of their own little networks, effectively. You could have a Slidester or something like Friendster. In any case, mediator is an ongoing project in the laboratory, and we're in the midst of doing a study where we've done every single mediator bait, and we're looking at it in a dynamic fashion with the Conaways. But the data is just being analyzed now. So we've been working on it for years. It's a very large complex involved in basically transmitting signals between activators binding enhancer sequences to get RNA polymerase II going. And in previous studies, here's a silver stain gel that you always know that people love to see these types of things. And here's this helicell extract. And I'll mention, I got a kick out of on Rob's slide, there's this 38 kilodalton protein serine threonine kinase that people in the mediator field thought was actually a kinase associated with mediator for a long time, but it just comes down in the anti-flag purifications all the time. It's such a simple thing to do. It's amazing how many people don't do these purification controls. So many years ago now, well, 5 years ago now, we did this study, and I apologize for the red-green, it's never been updated, where we had a series of baits. We're looking at sequence coverage across all of here of all these different mediator subunits. And then mediator pulls down pretty significant amounts of RNA polymerase II. And one thing that we noticed at this point in time is here's Med8, where we have a good antibody for it, and 2 different baits are down here. We're going to get 50% sequence coverage, which one's not 2 on the other, and 50% sequence coverage. It looks very similar on the semi-quantitative Western blotting. CDK8 is different, where we have 33 to 18. We have this difference, RPB1, where we have 41% to 18%. So we have this correlation of the semi-quantitative Western blotting with the sequence coverage. So we wanted to redo this study in a much more quantitative fashion, where we're going to do a label-free quantitative proteomic analysis of mediator. It shows 4 different baits from here to do 4 biological replicates and analyzed by MudPIT and NSAF and then statistical analysis. So here are the core components of metazone mediator, where the B's stand for the actual baits. Sometimes the cell made a massive amount of overexpression of it. Sometimes the cells did not. I don't remember the exact expression system in this case. But for the most part, no matter what bait we used, we only saw a couple cases where there were statistically significant differences. So each one of these little columns is the information that came from one of these 4 baits. So for the most part, we don't have this variation amongst the core components. And we did something like an N15 aqua type experiment to satisfy one of the reviewers to show that we're in a linear dynamic range. For this Med9 protein, we spiked in whole purified from E. coli and N15 and actually developed like a standard curve, for example. And in the case of one of the major interests in my group is to do these very large studies and start looking at protein complexes, say, during the cell cycle or during stimuli, which we'll get to at the very end. And in the transcription field, people will do this with some degree of frequency, but they have to have an antibody against everything. And antibodies don't react the same, even at the same protein level. There's just so much variance in the way they behave. So to validate some of this, we have Med8, Med17, Med18, where we have good antibodies, and we see across the 4 baits, we don't have any variance. Where we start to see variance are on these 2 modules that are actually functionally significant. So mediator has something called this kinase module. Now we've broken out, there are 6 proteins in the kinase module, and one particular bait that's called CRIS70 and Med26 has been renamed, has very low amounts of it compared to the other baits. And we can see that by semi-quantitative Western blotting, where we can't detect CDK8, and we can barely detect cyclin C, which is also in here. In the other 3 baits, we can see significant amounts of that. With RNA polymerase II, there's 12 subunits, named 1 through 12, going from largest to smallest. We're getting statistically significantly more RNA polymerase II associated with Med26 than we are with, say, Med29 and Med28. And Med10 and Med26 may have somewhat similar levels. We can validate that to a certain extent by the semi-quantitative Western blotting with Med26, with RPB1 and RPB9, which we have good antibodies for. We see the most of it in Med26, and we see diminished amounts in Med29, Med28. And then Med10 is probably somewhere in between according to the Western blotting. But what we do have is a case where Med26 is pulling down a form of mediator that has this different ratio of kinase module to polymerase module than the other baits that we're using. And this actually had functional significance in an assay strictly for transcriptional activity reflective of polymerase content, where if we add in purified RNA polymerase II, we basically get the same activity. But when we just add in the baits themselves, Med26 is the highest activity, Med10 had some, and the other ones have squintable levels of activity. So we have this actual functional significance of this quantitative analysis where this ratio and balance of polymerase to kinase allows for activity, whereas in other cases, the kinase module, in the case of Med10 in particular, we start to become competing against each other because this is thought to be repressive and this is thought to be an activative type of form of mediator. So this is a type of study that we're doing a fairly significant amount of these days in a lot of different contexts. And the second technology that we've worked on developing is this probabilistic assembly of protein interaction networks using spectral counting. And this was done on a chromatin modeling complex that's centered on TIP49A and B. So protein functions in complexes, and they're oftentimes part of multiple complexes, and TIP49 and B are no exception. They're rather promiscuous, in fact. And there have been other approaches out there that have been very valuable, but they've been binary, where basically you're saying presence and absence. And we know that there is more information than that. And also, the existing early methods, especially in the yeast work, were really relied on very large-scale data sets. And some of the math that was behind it couldn't actually, like this hidden Markov model, couldn't actually separate out known, separated, and distinct complexes. So we were concerned about that. So we developed a novel quantitative probabilistic strategy for assembling and looking at protein interaction networks. So in other work with the Conaway group, we've done a significant number of these work on this TRAPTIC60 complexes and other related complexes. And then you'll notice that there's certain baits that pull down TRAPTIC60, MRGBP being one, and these shared components, in addition to unique components of something called SR cap. YL1 does the same, but this zinc finger hit 1 and H2AZ are only pulling down the shared components, and the SR cap containing complex components, but not over here. So it's all centered on these two guys that are conserved between yeast and humans, this RUB1, RUB2 in yeast, 49A and B in humans. They are triple ATPases with roles in a wide variety of activities. They're conserved, they're in multiple complexes, and it's largely involved in chromatin remodeling in yeast, but that's not entirely the case in humans. So as we go through, we've developed we had all these baits, and we want to come up with a better way of analyzing this. And so in a very similar way to what Rob described, is we have a series of, in this case, an equal number of just cells passed through the affinity purification protocol to build a control data set. And then we have all the different protein complexes. So there were 27 baits, but there were replicates inside of here, so 35 total runs, and we had 35 total of these. The postdoc who did this computational work, Michał Sardiu, insisted that we have equal numbers of actual experimental to control data sets, and people are always really motivated to do a lot of control experiments. That one's always really popular, especially when I don't have to do it. At the end of the day, what happens is we get to this point where we have a data set that has spectral counts, and we can do a contaminant extraction by either subtracting out. In this case, we use something called the ratio of vector means, a vector algebra-based approach to subtract out information. Because when you do enough of these, you're going to have proteins that are going to be part of complexes, but they're going to be underrepresented. And things that come down in here at some point in time are going to be part of a complex. You'd hope that they would be overrepresented by spectral counts eventually. So then there's a series of steps that we take to basically get to a Bayesian, a probabilistic interaction network where every link is represented by a value. So part of this also was, in work we also published earlier this year, was looking at clustering more carefully using this value. And prior to this, a lot of proteomics had been done and clustering had been used, but it was still very binary. So we are interested in how can we use these quantitative values to do clustering. And we've found a couple of approaches that work with NSAFs and a wide variety of ones that do not. But it did a pretty good job of capturing and separating out. We have informative dendrograms. And that helped us to assemble this kind of Venn diagram of a network where you can find the core complexes, certain things that we're calling attachments, and they're called attachments at this time, that are not represented in every bait that pulled on a complex, but represented in some. And this is, all four of these complexes have this TIP49A and B involved in it. And there's this one protein that we're interested in mechanistically that only ever shows up when TIP49A and B are used as baits, but we never see them in any other case. So we're curious what that function is, but those studies haven't been studied yet. But the point of this was to use spectral counting for doing the probability of these interactions. So we're using just a very rudimentary Bayesian approach where we're going to generate a probability using the spectral counting, using NSAFs, using spectral counts, to attach an actual value to every single one of these linkages. And so in black in here are the highest probabilities, the highest numbers, and then down in yellow, which is difficult to distinguish from the green, are the lowest ones. So every single association, every single linkage in this network has an actual number associated with it. And the dark black lines are ones that have been validated before in literature. The dashed black lines are ones that had no pre-existing information about, but also had very high probabilities. So something that we're really interested in to this day in its ongoing work is, what do these numbers mean? So to begin, we kind of have to develop these two separate approaches. One that we're looking at kind of the abundance of a complex. Perhaps we're capturing the stoichiometry, but we need to do some more work to determine that completely. And then one where we're using to actually get these kind of, is it a strength of an interaction? What do these actual numbers mean? So let's look at one of the complexes. where we have, in this case, we just had 5 baits, and there's a significant number of proteins. We now have many more baits along this. We'll do an updated version of this one of these days. So we have a couple of these events where we have MRGBP interacting with DMAP. MRGBP is here, where DMAP1 goes here. It has an intermediate probability, and it actually has a co-IPable event. Here's a couple other ones. MRGBP with TRCP120, which is here, has a very high probability, and we validated it's a co-IPable event. Another one, MRGBP with YL1, which is down here. It had a co-IPable event and a probability of 0.01. So it's an intermediate. MRGBP with MRG15, where'd that dude go? Oh, here we go, right here. A co-IPable event and a relatively high probability. When we get down to this 0.00 type of range, this is MRGBP with TIP49A. MRGBP and TIP49A would be somewhere in this, where we go, up here at top. Is one of the lowest probabilities in the data set that was not co-IPable. So this got us interested in this question of what do these numbers mean? Do they actually represent associations, a strength, a direct interaction? Can we perhaps use this type of information to build a model of a protein complex, an architecture, if you will? It seems from the information that we have and the new information that we have is that MRGBP is the scaffold for this complex, but what do these probabilities, do they reflect the strength of the interaction or do they mean something else? This is where we decided to do this last part of the presentation on this protein complex, a specific protein complex in yeast where we can use genetics to study a deletion network. And we don't have an answer yet to this question, but we're getting there. We're starting to build up enough numbers where we have a feeling for it does in fact represent direct interactions. So again, here we have our RUBB1, RUBB2 in yeast, which is what we've been studying in humans with TIP49AB. So we kind of go back and forth between yeast and humans. Now we're going to do a study of this protein complex called RPD3. There's a small and a large form. There's 3 components shared between both complexes. And it's involved in chromatin remodeling. The small complex is involved in suppression of cryptic transcription, and on the RPD3 large complex is in more just in general repression. So we have 2 different types of affinity purifications here. One is basically all the baits have been tagged and analyzed. And the other one's going to be where we're going to have one of these baits at this point in time where we're going to go in and have the RPD3 tagged, for example, but we're going to have a deletion of something else. Genetically, you're going to remove DEP1, or we're going to remove RXT2, but we're going to do the affinity purification. And we have also the SYN3 tap with deletion of RPD3, but that's not as useful as having kind of the systematic almost of all the other ones. It obviously depends on something being viable versus lethal. And fortunately, the majority of these, actually all of them, are viable in the large complex in yeast, so we can do these experiments. We're using the same kind of general outline, but what really was most informative was the clustering of the actual deletion interactions, more so than, say, the actual probabilistic network, where we're able to kind of use the logic here to start to build up this kind of interdependency, who's related to who and who's dependent on who. So the wild-type network itself, when you're looking at a small complex, you just have a lot of very high probabilities. It's not particularly informative. In the deletion of the SYN3, in the case where we have where SYN3 has been deleted and we're doing the RPD3 tap purification, basically, we go from having color to red. The complex basically falls apart as far as we know at this point in time, where SYN3 seems pretty clear to be the scaffold. So SYN3 being the scaffold, but is there other architectural information present? So we have the wild-type purifications that don't really show us very much. We see things cluster with each other. The small complex unique components cluster down here, the large do here. But the deletion case, where we have RPD3 tap and then all these components have been deleted and analyzed, turned out to be much more informative. So here again is the wild-type purifications, and all we're really doing is getting separation of the large and the small complex. In the case of the deletions, though, we're starting to using the dendrograms to say what are the submodules, what's the organization and the rationale perhaps for this complex architecture. So let's walk through it. UME1, RPD3, SYN3 remain in all the deletions except the case where RPD3 is deleted or SYN3 is deleted. These are these core modules in both complexes. They're very stable in all the deletions. When we look down here at just the small complex components, in the case when EAF3 is deleted, obviously EAF3 is gone, but RCO1 remains, is still detectable. When RCO1 was deleted, both components, RCO1 and EF3, were no longer detected. So it seems like RCO1, well, say EF3 is more dependent on RCO1 for being part of the small complex than vice versa. So perhaps we can propose some sort of organization like this. When we go to the large complex, there are these sequence-specific repressors that are present at kind of substoichiometric and lower abundance amounts. So here's the case of ASH1 and UMA6 in the deletion, where when we delete CTI6, we're losing CTI6, ASH1, UMA6. The UMA6 one, we still have ASH1 present. ASH1 deletion, we're losing everything. So it looks like we have CTI6 when it's deleted, which is a key component. It's a stoichiometric component of this. Not much else happens except for ASH1 and UMA6. The RXTs, FO23 are present, SAP30, DEP1, STS3 remain. In the FO23 deletion, all we're losing is RXT3. So we're losing RXT3 and FO23, RXT2 is present. These other components are still present. In the RXT2 deletion, RXT3 and FO23 are lost. So this suggests that, this suggests that RXT2 is perhaps more important. I'm sorry, FO23 is perhaps more important. We'll see that later. Take a little bit of a perspective, different perspective though, on these kind of core components that are present in many cases when we start deleting them. So when SAP30 is deleted, STS3 remains, DEP1 and SAP30 obviously, DEP1's lost, SAP30 is also gone, and then these other ones, FO23, RXT3 are gone, but RXT2 still remains. In the DEP1 deletion, all we have left is RXT2. All these other components, we still have the intact core, all these other components are gone. In the SDS3 deletion, all we have left is the core. We have this long amount of of black. Things are just gone. So when we use all this information, can we start to build kind of rudimentary, at this point in time, architectural models of protein complexes? Is there a way to use this to kind of come up with a structural type of model of complexes? If we had the actual crystal structure of all these individually, could we then build a better looking version of this? And that's an open question at this point in time, but we're using this type of an approach very widely in the laboratory on human mediator, also on other yeast complexes, to start to get this type of organizational type of an approach. We also want to go back in and to a certain extent, if I can get funding for it, to go in and be much more systematic, where we do this, rather than RPD3 tap and all these deletions, we say do SYN3 tap and all these deletions, UME1, to be much more systematic and get the whole coverage to see, can we start to also ask questions about how complexes are assembled in vivo, taking this type of an approach. So today I mostly talked about work that we use in the laboratory for this normalized spectral abundance factor. We talked about this quantitative proteomic analysis of mediator, where we had these insights into different forms of mediator and perhaps the stoichiometry of the complexes. We do probabilistic modeling of interaction networks using spectral counting. We have a very large study that I'll start talking about probably early next year on mediator. We've done all the components in a somewhat of a deletion, non-deletion type of work, similar to down here. And we're interested in can we use these perturbed networks to start asking questions about how things are assembled. You could imagine using a similar type of approach here where, say, you use a drug instead, and using the clustering type of approaches to see how these protein complexes change. Here's a picture of the Stowers Institute. We are up, which one? Up, yeah, we're here on this third floor. And my wife and I co-run the group. Both of us trained with John Yates, Laurence Florent. Andy Paoletti did the work with Ron and Joan Conaway, who are really outstanding collaborators with us on the mediator work. Mihaela Sardiu is our really outstanding computational proteomics and interactive network. She's like our one-person Interactome group doing all this data analysis, using a lot of the data we've published about in other biochemical ways from the Conaway lab. And Josh and Mihaela worked very closely on the RPD3 Syn3 work in collaboration with Jerry Workman, who does the biochemistry of RPD3 Syn3. With that, I will stop. Thank you for your attention. I'll be happy to answer any questions.
Give a little perspective and comment for the group about the use of ion current values as compared to spectral abundance, and what are the advantages and disadvantages in various contexts?
So we find on the, the instruments we use are the LTQ and the LTQ Orbitrap. So the data sets that we have, and we have work that we're working on, unpublished kind of analytical chemistry work that we've done to do those things. What we see, especially on the LTQ, say, versus the previous version of the instrumentation, that spectral counting has a wider dynamic range, close to 4 orders of magnitude. And ion currents start later, and spectral counting will start to saturate earlier than we do with ion currents. So they're kind of on different types of scales, where spectral counting can start here and starts to saturate. Ion current can go here, go up a little bit higher, but doesn't have the same dynamic range as spectral counting does. Certainly on an Orbitrap, having the good resolution for endpoint picking is essential. You can't do ion current well on an instrument that doesn't have the resolution of an Orbitrap. It's previous life, I did a lot of ion current analysis using a DECA, and the problem is endpoint picking. If you miss this much of the base, your errors are way off. So I think it's very instrumentation dependent. I personally think spectral counting is an easier and has, especially since mass spectrometers are constantly being developed to be better and better at identifying peptides, I think it'll be a dominant quantitative approach pretty soon. For interactome network analysis, it's not really realistic to spike in N15 something every single time. The cost becomes very large when you do those types of things. And so we're really focused on label-free spectral counting-based approaches because I think it's what we'll need to use to do the types of thing that people in this room want to do. That's a long-winded answer, but it's my answer. Please.
So I really like the way you're able to sort of deconstruct those complexes, and I was wondering whether, the core components, do you think they represent, is there any sort of, if you like, chronology there? Could you think you'd be able to find, the core components, if you like, in complexes of their own before other proteins are?
I think that we have unpublished work that we're doing in Mediator right now where, the other thing that you could do with this type, the beauty of yeast genetics is that you can go in and we could put in a GFP tag in a deletion background, and we can start to see, say, using things like FRAT or FRAP, in vivo, is there a core formed when these other components are deleted? And those are the types of studies that we want to start to do. We believe that that'll be the case, that it's reflective of the in vivo assembly of complexes, but we don't have the data to really, the proof beyond a reasonable doubt yet to say that. But I believe that that will be the case. And Claude, please.
Do you ever monitor the abundance, after you do your deletion experiment, do you monitor the abundance of the protein in the lysate prior enrichment? And if so, in what kind of frequency do you see actually the abundance of the protein being affected by deletion of one of their binding partners?
That's a good question. We haven't done that at all.
We see it a lot, so it'd be nice for you to know for your.
So prior to the purification step, what are the abundance of the proteins in the cell? No. We haven't done that yet.
How reproducible are the peptide identifications? You see the same peptides for the same proteins over and over again through multiple runs? Or do you see different?
Pretty much. We've done a big RNA polymerase 1, 2, and 3 study to start to try to address that this other postdoc, Amber Mosley's working on. And especially when you get the things that if you see something like with 10 spectral counts or above, those become very reproducible. In protein complexes, the things that are 1 or 2 peptides that you see as 1 or 2 spectral counts, those might fluctuate. But the ones above a certain number we see very reproducibly, especially these things that you get hundreds of. So it's the peptide level reproducibility is actually quite high. It might be higher than the protein level reproducibility because when you get down to those kind of serendipitous proteins that show up with 1 peptide, 1 spectral count, they wax in and out of existence. But when you're dealing with these thousands, the things that vary for the individual proteins are the ones that are very low. It turns out to be much more reproducible than I thought.